Hello everyone, my name is Yejun Kim and I'm a senior in the Holmes lab where we study yeast genetics and epigenetics. And so a little bit of background about my project. We know that DNA in eukaryotes is organized into nucleosomes, which is composed of a core protein complex made up of 2 copies each of 4 histone proteins. And also the DNA winds around this core complex to create the nucleosome. 2 other proteins of more interest to me in my project specifically are histone H2A.Z, which is a variant of histone H2A and replaces H2A in the nucleosome by the action of the SWIR1 chromatin remodeling complex. And also histone H1, which is the linker histone present on the linker DNA outside of the core protein complex. So we know that nucleosomes interact with each other to help compact DNA and alter chromatin structure. And also a lot of post-translational modifications occurs for these proteins to affect things such as yeast gene expression, chromatin structure, chromatin dynamics, etc. And so my project specifically wants to learn a little bit more about the functions of histone H1 and H2A.Z. In the lab, we know that when you delete histone H2A.Z, a lot of defects are caused in yeast cells, such as in telomeric silencing and defects in chromosome condensation. And alumni members in the lab have found that when we additionally delete H1 along with deleting H2A.Z, the defects caused by deleting H2A.Z only are suppressed back to wild-type levels. And so now with my project, I want to ask if the deletion of H2A.Z causes any other defects in yeast cells, such as in non-homologous end joining, which is the pathway by which yeast cells repair DNA double-strand breaks when there's no homologous template to work off of. And also, are there any defects caused in plasmid retention, which is the ability of the yeast to retain the plasmid and pass it down onto future generations? So sneaking forward and a little sneak peek into my project, we have found these defects, or we have found that deletion of H2A.Z causes defects in these 2 processes, and the relation with H1 can be further found in my poster, and I hope to see you there. Thank you.